Hello, everyone. Okay, hi. Okay, I'll just give everyone like 10, 15 seconds to settle down and then I'll get started. Okay, hi everyone. My name is Anmol Singh. I'm a recent graduate of the Bachelor's of Science in Bioinformatics program here at Langara, and I'm honored to be speaking in front of all of you today as well as with my panelists. I'm excited to hear their journeys. I'm excited to share my journey, and I'm going to start off with a diary of a DNA inspector. You might be wondering why DNA inspector, and it's because whenever my dad introduces me to people, he says my daughter is a DNA inspector, not knowing what I do. So that's why this is Diary of a DNA Inspector. The journey to discovering bioinformatics. So my story starts off at 2 years old when I was fully determined to start programming on my toy computer. So that's me at 2 years old doing that. Unfortunately, it didn't pan out, but there was some foreshadowing, which led to me to my bioinformatics here today. So in 2017, I was fully determined to go to Langara, transfer to a different institution, pursue med school, and become a doctor. That was my goal. That's what I wanted to do. I was fully determined. Around 2019 comes along and they tell me about this bioinformatics program, this bioinformatics thing everyone's talking about. I wonder what it is, and turns out that it's an emerging new field. It's only about 30 years old. There's a lot going in it. So I was like, oh, let me explore this. Let me see what it is. I ended up transitioning to the bioinformatics program, and it was easily one of the best decisions I made. And I recently, this year, completed my Bachelor of Science degree in bioinformatics, and I'm a recent graduate. I've gotten a lot of opportunities through the bioinformatics program, which I will share with you today during my career history. So my first exposure to bioinformatics was with a company called Microbiome Insights. I worked there as a co-op student. So Microbiome Insights is a company that performs bioinformatic analysis for microbe-related or microbial-related environments specifically. So what they do is you provide them with a sample collection. They will sequence it, they will do a sample processing, they will do the bioinformatics application, and they'll design a custom report for each client. I did not do any of that. Instead, I was a database development intern. So I was actually working on a special project in collaboration with Health Canada, where we were going to design a mega database. The goal was to design a database that encompassed a lot of microbial databases. And so the portion that I worked on was first developing the SQLite version of the NCBI taxonomic database. So once that was done, we then integrated the databases Backdive, Patrick, and Uniref. The great thing about this experience was that it actually, first of all, it allowed me to dip my toes into what bioinformatics is. I got to experience it firsthand. I saw there was a different application of it. It wasn't just biology now, it was also the computer science aspect where I'm working with databases. I also got to write Python and R scripts for the first time and see what interfaces are like. I worked with an API for the first time ever. And I even got a chance to prepare documentation and explain workflow through the documentation. So it was cool to see everything from beginning to end. It was also, again, one of my first time stepping into bioinformatics, got to see what I liked, what I didn't like, where I wanted to go, and provided me with a more full view of what bioinformatics is. I then got an opportunity to work on the cannabis sativa pangenome project. If you don't know what a pangenome is, that's totally okay, because I'm going to build your vocabulary. So to start off, everyone or most people know that DNA is the molecule that carries genetic information, but we also have a genome, which is the complete set of genetic information in an individual. So everything from beginning to end is located within your genome. There's something called genotype, which is the genetic makeup of an individual within a species or a population. So everyone's genotype is unique. Everyone has a different one. And there's phenotype, which is the observable trait that determines your genotype or is determined by your genotype and by environmental factors. For an example, eye color. My eyes are brown. Why are they brown? Because of my genotype. It codes for the brown color. The phenotype is the observable trait. You can see that my eyes are brown, and that's because of phenotype. So a pangenome is a compilation of multiple reference genomes. So it's using all of the genomes that are available in that species to provide an understanding of all the genetic diversity in that species. So on your right side, you will see that there's 3 organisms in this species. The areas where the organisms will have unique components to them, but there's also areas of overlap. That means those two reference genomes will align. That's the area that they share, a sequence that they share. And then there's a sequence that all of them share, which is located in the core in the middle. So the pangenome is everything, whether it's shared, whether it's not shared, all of it all together. And the great thing about pangenomes is they allow us to understand the genetic diversity that occurs within these populations. So what our goal was with the cannabis sativa pangenome project was to first of all construct the pangenome, but also to connect phenotype to genotype. We wanted to understand why certain sativa grew a certain way, or why certain ones smelled a certain way, why one of them had this feature and not that feature. So the entire point of this was to use all of the genomes that were available to us, construct them, and see what we could find, what we couldn't find. My responsibilities and duties were to research to determine the best algorithms that we could use. So I was the first step. All I would do is I would research, figure out which one was the most efficient, which one was the best for us to do. The thing about plant genomes is that they're very complex. So the previous people who had constructed pangenomes, we couldn't necessarily use them the same way because of how complex plant genomes are. So it was a lot of trial and error, figuring it out, and then passing it on to the next person. This also meant I got a chance to write documentation, and I got a chance to write procedures to see kind of how it was going to go. So yeah. We actually got a chance to present our research. That's my friend Mike. He's in the audience as well. I made this poster. I love making posters, so I was really grateful to do it. And the chance to present our research was also really great. You can see the pangenome, which is located small, but that was our first attempt. We had 14 assembled genomes. We aligned them together, and that's what came out. So it was a really good step to discovering the cannabis sativa pangenome. It was also the very first attempt in all of human history. So it's pretty interesting and intriguing. And it's crazy to think that a bioinformatics degree allowed me to do something for the very first time in human history. I then transitioned a little bit away from the bioinformatics side, and I went into something called the Applied Research and Technology Project. The purpose of this project is that they have other companies provide data, and then you use the data that's provided to help the companies with whether it's like reducing operational costs, to reduce wait times, or just managing and analyzing their data. The purpose or the way that we achieved this was through designing prototypes, systems, and algorithms for these companies. And our industry partners included Springboard Clinic, HeadCheck Health, Citus Insights, and Life Labs. I was working as a junior data analyst, and I worked on two projects specifically, one with HeadCheck Health and the other with Citus Insights. So with HeadCheck Health, if you haven't heard of the company, they basically partner with bigger organizations such as the CFL or NFL, and especially sports or companies that have a lot of concussions. And what they do is they help the athletes determine data and statistics. So my role in that was given a set of data that they had provided us with. I was to write an algorithm and ensure that it could be used in the future based off of the questions that they had provided us with. So there were a certain set of questions they wanted us to answer. So I was writing these algorithms. I had to make sure that it was transferable for future data as well. And in the end, I got to create reports and generate reports in R as well as Superset. So just to show them a little bit about what we're doing, what they can achieve from the data that they have provided us with. I also got a chance to set up the Google Cloud to host servers with data and for data and prototypes, which was also really interesting because at the time I was learning about cloud computing, so got to apply that right away. At Citus Insights, we were working, well, the company basically is a health-related information technology company, so they do data analytics and logistics for other companies. What we were trying to do was research, develop, and implement a data scoring algorithm for them. And my role specifically was data analysis. So I wanted to make sure that the data that they provided us with and the data that we had transformed was statistically similar. Here I was using machine learning algorithms, another class I was taking at the time. So it was pretty cool to see the skills that I was learning in school were directly transferable to the bioinformatics program or bioinformatics in general. The other great thing about this is this job is not bioinformatics. This job is actually data analysis. But because of how vast the bioinformatics industry is, I was able to apply the skills I had learned from bioinformatics into a job that has nothing to do with bioinformatics whatsoever. So it just goes to show how vast bioinformatics really is and the great things you can really achieve with it when you set your foot or you decide to try new opportunities. Present day, I'm working for the Langara Bioinformatics department. So over the summer, I got to do some course development where I created a summer, or I helped create a summer camp program for high school students that would be interested in bioinformatics. This was really interesting and really fun because one, I got to pretend I was in high school again, but it was great because I got to figure out which core concepts I really wanted to introduce to students learning about bioinformatics for the first time. And I got to see like how I wanted to create these lesson plans, how I wanted to create interactive activities, what I wanted to introduce. So we decided to focus on viruses, mutations, phylogenetics, and genomic epidemiology. Present day, I'm working as a student advisor. I focus on student recruitment. I go to different classrooms and I do presentations telling them about the bioinformatics program, what's great about it, what we love about it. I also provide guidance to students who are, as in like helping them with courses and where they want to do their co-ops and stuff. And I also participate in conferences and career fairs to educate people, educators and students on bioinformatics and what we have to offer here at Langara. So that kind of concludes my career journey thus far. It's been great in the sense that I've been able to apply everything that I've learned in very different areas of my life. I worked in database development. I've worked in actual bioinformatics with the Cannabis Sativa Pangenome Project. I worked with data analysis, and now I'm just doing fun talking things, which is also really great. And it's crazy. Bioinformatics gave me that opportunity. And even just standing here in front of all of you today, like bioinformatics did this. I'm grateful for it. There's also some personal projects that I've worked on in my own time. The first is PhyloMe. PhyloMe generates phylogenetic trees. If you don't know what a phylogenetic tree is, it basically depicts the evolutionary relationships between species. So for an example, if you look to the phylogenetic tree on your right, you'll see that you have present-day species and ancestors. So species A and B are closely related, species C and D are closely related, and species E serves as an outgroup. That means A, B, C, and D had a more recent common ancestor than all of them combined. So this is really cool. And the way that I applied it was I decided to use the TP53 gene. If you know about the TP53 gene, you know that it's a gene that acts as a tumor suppressor in many tumor types. TP53 is actually a gene that has mutated a lot over time just through evolutions. It mutates very easily. And the other thing about it is in humans, it actually represents itself very differently depending on the cell type. So it's a really big marker for cancer research. What I did was I constructed my phylogenetic trees. I saw that I used the unmodified TP53 sequence and the modified one, and they actually depicted completely different trees. It just goes to show that at a cellular level, they depicted different relationships, which is an important marker for when you're making drugs, for an example. You're making the medication or you're helping therapy. So it's an application, and I was able to apply it from bioinformatics. The next app that I made is Mahan. This one is named after my father. It's the Mind and Health Aid Network. This is an app I made for caregivers because I'm a caregiver. And the whole point of the app is to help caregivers make their day-to-day lives easier. So I included and implemented a appointments function, a medical records function, and just helping people keep track of medication, appointments that are upcoming, surgeries that they've been to. And the reason why I present this app is because it was done on my own time in a programming language I had no experience in, which is Swift. I don't even have JavaScript experience, but because of bioinformatics and the programming languages I had acquired over time, I was able to pick up on another programming language. And it just goes to show that even though I had pursued one field or one area, I had been able to apply that and create something of my own. And next, I'd like to mention some aspirations and some challenges I faced. So my aspirations, short term, is to attain industry experience, which is code for get a job. So yes, currently in the process, looking for a job. And I really do want industry experience and just to see what that's like. That's a short-term goal, working towards it, applying where I can, going to different events. But that's a short-term one. Long-term goal, as I gain more experience and gain more information, I really want to bridge the gap between bioinformatics and the South Asian community. As a South Asian myself, I know which health issues particularly my community faces, and I really hope in the future that I'll be able to provide them access with bioinformatics tools and knowledge just in general to help bring the community healthier, you know? and some challenges and how to overcome them. So the first challenge I faced was developing a coding brain. I went from a biology degree to computer science for the very first time, and it is hard. Becoming a bioinformatician or just going into coding like with no experience means that you need to learn how to think like a computer. It means you have to rewire your brain completely. You have to understand why this if-else statement works and this one doesn't, or why is this code running this way and not that way. And the great thing is, with time, with practice, you can develop your coding brain. So utilizing online resources like LeetCode, HackerRank, even AI now, everyone has AI. You can access AI, use AI to teach you, to give you tips and suggestions. Another thing that I think a lot of people face is imposter syndrome. This idea that you're not good enough or that you fell into where you are by accident, by mistake. But the reality is you have to remember you worked really hard to get where you are. There's a lot of history. There's a lot of things that you have accomplished, and you were the one who got yourself there. So why feel like you're an imposter? You're the real deal. So I think that's an important thing to remember as well. As of right now, based off of the job market and the way that things are, sometimes having a lack of network can be a really big hindrance. And it's important to build connections. So a great thing that you can do is attend networking events like this one. VanBug is great. There's also other networking events and opportunities you can go to, or reach out to people on LinkedIn. One thing that I would suggest to everyone is learn how to talk. And that means be able to talk your way into opportunities. And you never know, when the more you talk to people, the more opportunities people will present. So there's one thing to take away: learn how to talk. The other thing I would like to say is online application systems can be really difficult too, and they can actually be pretty demotivating when you realize that, hey, I may not be the first, I may not be the 10th, I'm actually the 100th or the 1,000th or the 10,000th. But the thing is, when you are applying, you have to have hope. You have to keep that hope, and you have to be like, oh, I can do this. I will be able to do it. When I apply, I make sure to use as many keywords as possible from the cover letter or from the job description into my cover letter and resume. That way, I'm more likely to get a hit because they're using AI just like I am. So important to be able to work around that. So use as many keywords as possible. So yeah, that concludes my talk. Thank you so much for listening to me today. There's some people on the screen, their names have been overlapped for some reason, but shout out to them. If there's any questions from the audience, I'd be happy to take them now. Thank you. No. Well, I'm glad you guys like my talk so much. No one has questions. Okay, then I will hand it off. Thank you so much.
And next we'll have our speaker, Dr. Lau.
All right. Hello, guys. My name is Venus, and thank you for having me today, and thank you for the invitation from the Vanbug team. It's nice to see a lot of familiar faces and also new faces that I might meet later. So I'll be talking about sort of my journey in navigating a more non-technical career in bioinformatics, particularly down the industry route. And when I say non-technical, I mean like R&D or engineering type of position. So just very quick background, I completed my Bachelor of Science in Molecular Biology and Biochemistry at SFU. And during my undergrad, I was also able to develop some co-op experience in the wet lab, ranging from working in a tuberculosis research lab at the BCCDC to cancer immunology research at the Genome Sciences Center, so which sort of conveniently helped me land a job as a research technologist at Stem Cell Technologies right after I graduated. So I spent around 2 years there focusing on developing reagents, like clinical grade reagents for CAR T cell manufacturing. And my day-to-day life during that job was to spend a lot of time in the lab doing like cell purification, cell culture, like flow cytometry analysis. And after doing that for 2 years, I sort of wanted to consider a career change. Like I still want to stay in science, but maybe move away from the lab bench. So that's when I started to think about, grad school. And I was able to get a PhD position at with Dr. Fiona Brinkman's lab at SFU through the joint bioinformatics graduate training program between SFU and UBC at that time. So during my PhD, so when I got into the program, I actually started with no experience in coding, and I didn't even take any courses in bioinformatics before that. So initially, my main focus was to really quickly build up like programming skills to get my project started. So my thesis work in the Brinkman lab, so we do microbial genomics and bioinformatics research. And my particular project was to develop a computational workflow for identifying and characterizing bacterial pathogen-associated genes that could potentially be used for antivirulence drug development as part of the efforts to combat the growing threat of like antimicrobial resistance. So just like any bioinformaticians or computational biologists, I developed, the basic like technical skills from coding and troubleshooting, from, learning how to run on the command line and on the HPC like Compute Canada, building bioinformatic analysis pipelines, analyzing those data and presenting those results visually. And that also includes, being able to present the data through presentations at conferences, thesis defense, to presenting those data in writing in the form of like peer review articles, even scholarship applications. And these are some of the skills that I still learn today, but in different form. Like for example, instead of writing articles and scholarship applications, now I spend a lot of time writing business grants, talking about our, I'll talk more about that later, but yes, like writing more business grants and also doing a lot of like product demos and doing other types of presentations that are not so research focused. And besides working on my thesis, I also had the opportunity to get involved in a lot of, bioinformatics community involvement, including, the SFU Omics Club with my friend Michelle here, the MBB Graduate Caucus at SFU, and also Vanbug, which I spent, I think, 2 or 3 years with the team. So it's just nice to be back here. And through these experiences, I was able to develop other soft skills that are also crucial to my current job. And these include partnership developments, working with leaders from different sectors, to, get event sponsorship, funding, and also helping them with like job announcements and advertisements. And also like event organization, from organizing monthly seminars, workshops, to career panels and conferences. And also, so through these process, I really developed the skill to, learn how to network professionally with people that could be a lot more senior in the field, and just building up all these like academic and industry like connections that could potentially help me further down my career. So, like with my technical skills in bioinformatics and the other soft skills that I've developed, that really positioned me well to transition into a non-technical job within the bioinformatics space. So I now work for an early biotech startup based in the US called Magna Labs. So what we do is we provide software solutions to help computational biologists or bioinformaticians to improve their analysis pipelines and results through like benchmarking, testing, and validating their analysis pipelines with like automation tools. And at our company, like we really focus on like research data quality, including like output consistency, like result accuracy, and data reproducibility. And so my role at this company is custom solutions lead. Yes, I know when I applied for this job, I didn't really quite understand what this job is about because it's kind of vague, which now I understand like why it makes sense, because in a small startup, I do everything from, like internally facing, working with our product, like engineering team, to like, externally facing, working with the community and with our like end users and customers. So I do everything from like product R&D, like implementing, like suggesting and implementing new features based on like user feedback and the trends, like research trends, and also helping with the engineering team with like bug detection and like pre-release testing before we deploy our software products to our users. And with that also kind of involves, product marketing. So when we have new features and new software products, like how do we bring these tools to the market, to the community? So I do a lot of like market research, like content generation, and like branding and positioning, because again, we are a very small startup. So, being able to gain exposure in the research field is a big challenge for us. So we've just been like working through like different strategies, seeing what works and what not. And that really ties into like the community engagement work that I do. So I do a lot of interviews and sales calls with people across like academia and industry. And a lot of times, like I'm not even trying to actively sell to them. It's really just to be able to have a conversation with people in the field, see what kind of challenges they are facing in their bioinformatics research, pipeline development, and see if we can offer any solutions to their challenges. And on top of that, I spend a lot of time doing like in-person or online networking and going to conferences and doing presentations. And through that, we find people that are interested in trying out our products. And then I would be involved in the user onboarding and user support process. And through that process, I also like listen to our current users, like what their requests are, or any customizations that we need to do to sort of bring our products to match their like bioinformatics applications or testing needs. So as you can see, my job at a startup can be like quite dynamic, and sometimes I would focus on like product R&D over community engagement, depending on sort of like the sales cycle that we're at, and depending on if we have a lot of users, if they're experiencing, if they want to scale up their developments in-house, like we kind of need to put in more resources to help them do that before we can spend more time, acquiring new users or new customers. So it kind of keeps my job really fun. And to be honest, like these areas of tasks that I do, it's still relatively new to me. So these are things that I haven't really gotten any like formal training in through grad school, through my bioinformatics training. And it's just really, it just involves a lot of like trial and error and like learning on the spot, which, I'm still growing and learning. So I was asked to talk about, some of the difficulties and challenges that I faced throughout my career. And for me, I guess the biggest challenge was to, transition from grad school to back to industry. And for me, because I knew I didn't really want to stay in R&D as much as I love the research, but I prefer not to do that full time. So my two main questions that I had was, what type of non-technical jobs in bioinformatics are out there and what companies I can apply for. So in terms of the job types, as you can probably tell from, my current role, there are a lot of things that you can do. So on the technical side, when you have a bioinformatics degree, the most straightforward career route is to continue in R&D as a computational biologist or bioinformatics scientist if you, enjoy the biology side of the research. Or you can also go into pipeline development if you're more passionate about, software engineering and development, and you can work as bioinformatics developers, engineers, data scientists, and there are also like emerging roles like, machine learning scientists have been popping up a lot in the last few years. And, bioinformatics test engineers that I often work closely with. And on the technical side of things, like if you don't want to spend a lot of time coding or doing research like me, you can go into things like product management, project management, like IT, specifically on the InfoSec and privacy side, especially if you're, developing bioinformatics software that you could potentially sell it to other companies or use it in like clinical settings. Security and privacy is very important. And you can also go into like sales, marketing, business development, or consulting, still using, all the skills that you learn in bioinformatics. So there's really an endless opportunities that you can explore. And it's just a matter of, being able to find the right resources to be able to like, learn the skills and apply to roles in these different settings. And in terms of like companies that you can apply for a bioinformatics related job, like life science companies doing omics is the most straightforward answer. So this includes like biotechs and pharmas that are developing clinical like genomic diagnostics, therapeutic development, or even like direct to consumer DNA tests like 23andMe. And on the right here's just an omics solution provider market map that has been developed and published by Latch Bio, also another bioinformatics company based in the US. So as you can see, there like omics related companies, like there are lots. These range from, like sequencing service providers to kit providers to instrument providers across the various like omics disciplines that, you might be familiar with. And it's also interesting to see that in the last year or so, these are just a few of the companies not in biomedical sciences that are also expanding their bioinformatics operations and R&D. And these include your favorite tech companies like Microsoft, Google, Amazon, NVIDIA. And if you're an animal lover, there are also a ton of like animal-related biotechs, including Embark. Like I think they provide like DNA tests for dogs to see what breed your dogs or pets are. And then like a gut microbiome company for pets, Animal Biome, and then an I think it's an epigenetic-based like pet age test company, EpiPause. And then if you are a pet owner and you go to your vet often, you're most likely familiar with, Hill's Pet Nutrition. They develop and sell like pet food. And they actually have a bioinformatics team that are looking into, the impact of nutrition on like gene expression in pets. So, it really depends on what you're interested in. And there's a lot of places you can apply for. And on the far right, you also have like agri-foods company that are using genomics tools for like crop improvement. And even for like berry breeding that I found was pretty interesting. So there are actually a lot more companies than what I've shown here, but these are just some of the companies that, I saw them hiring for bioinformatics, like scientist or engineer positions in the last year or so. So I would just like to wrap everything up with a few career resources that I personally found useful. So obviously, like LinkedIn is a good place for looking at job postings and also for networking. And that's actually where I applied for my current job over there. And if you have a big enough network on LinkedIn, sometimes you would also see like hiring teams or recruiters advertising these job postings on their personal profile. And sometimes you'll see that, feel free to reach out if you have any questions. So it's a good, invitation for you guys to like get in touch with people working in that company that you wish to apply for. And the second research, which can be debatable and definitely take it with a grain of salt, is the biotech subreddit. And I like to just scroll in that Reddit like every few days because it's really a good like informal resource to get like an insider news into like, the current job market and trends. And you'll hear experiences from, people that are applying for jobs, from current employees that talk about, the whole like interview process, like job application to, like the work culture at the company that they're working for. And, and I think this year they also had a like salary survey where you can actually like report your own salary anonymously. And there's a big like Google sheet that I can see how much people are making, like their qualifications and where they're working. And I actually find Reddit to be a pretty good resource for tracking company layoffs, because a lot of the times I actually find, I was actually able to like know which companies are having layoffs on Reddit before I actually see the same type of news on LinkedIn or in press releases. So it could be another good resources. And last but not least, like networking groups, whether it's in-person or local networking groups like VanBug. And there's also another group called Bioinformatics that's like a networking group based in Vancouver, and it's more for like general like life science like professionals. So you'll have people from, bioinformatics, non-bioinformatics, research, non-research. I think I've also met people from like commercial real estate that are helping like labs rent space, and also like lawyers, like angel investors. So, people from all sorts of careers. And also, I think I also recently found this online community called Bits and Bio. It's mainly based in the US, but they also have a very large online presence. And they organize a lot of in-person and online networking events. And they also have a big Slack channels where, people talk about, their current job, or, if they want to advertise for like new job postings, or if they're like a startup advertising their products, like you can kind of find any sort of discussion through these resources. So definitely take advantage of them. And that's it. Thank you for listening. Yeah. Feel free to ask any questions, or feel free to reach out to me through email or on LinkedIn afterwards too. Yeah.
So you mentioned the skills that you acquired, right? Was there any like memorable experience that you drew from in order to tackle some challenges you faced in your job?
Right now?
Yeah.
Well, I guess the skills that I developed that is still useful now would be like I would talk more about like the soft skills in like partnership development, like reaching out to different organizations, different companies. As a grad student, asking for like event funding, which is kind of similar to my current role, because right now I do take on a more like business development sales role where I actually need to go out, like reach out to companies and find our prospective buyers. So it's really the same skill, but you're just sort of like communicating different things, and you're like sharing different information with these different organizations. So there's a lot, I would say throughout your like formal training or even just like through life, your different activities, extracurriculars, like if you think hard enough, you can definitely transfer a lot of the skills from your, experience into like any jobs pretty much. Yeah. Yeah. Right now. So I'm actually lucky enough to be able to work for a startup that really values work-life balance, because I think there's that common misconception that, startup life is always like chaotic, which sometimes is. But I would say the most important part is to, like, whether you want to work for a startup or not, like, being able to understand what their work culture is in the particular lab that you work for or company. And these are things that, you can ask questions during the interview process to get an idea of what it will be like, or you can, reach out to existing employers and see, if you're going to be working like 12 hours a day on a weekend. And so like for me, I feel like I was lucky in that sense. And because my current role is completely remote and we are a fully remote company, so we get a lot of flexibility in terms of our working hours, as long as we show up for our meetings and get things done. It really doesn't matter when we work or where we work. So like earlier this year, I was able to spend like 3 weeks in Italy, where I was just working full time and traveling at the same time. So, remote jobs like this could offer that flexibility. Yeah.
Other questions? Thank you, Dr.
Yeah.
Lau.
Thank you.
And if you guys can also join me in welcoming Dr. Jackman. You want to move this? The question? Can do. Thank you. Yeah.
Hello all, my name is Sean and I'm a science geek. So I'm here to talk about my career trajectory, and I spent the first 7 years of my life as a Lego architect and technician. And my career went from there into the digital revolution. So I applied for a small startup grant from my parents to purchase an Apple IIe computer and went into software development and started a small boutique video game firm. The clientele was very limited, my parents and my siblings, but they were diehard fans. And that's where my programming experience started. So with the revolution of the internet coming, I upgraded my Apple II to an Intel 486 and got on the internet and got into web development and learned C and C++. And continued to enjoy programming and wanted to learn more about it. Coming into undergrad, I joined computer engineering at Simon Fraser University. And there I was convinced that the best career trajectory for me was going to be a video game developer, having spent most of my teenage years playing video games. In university though, through my coursework, I found that I really enjoyed the embedded firmware programming coursework, project coursework specifically. So this is programming where the programs live inside real tangible devices that do things. I found that applying programming to something that affected the world around you, just super interesting. So video game development was not in fact for me. And through the co-op program at SFU, I was able to get a job in that field. And I designed lighting electronic control equipment for the entertainment and theater lighting industry. So these are like the moving lights and things you see at rock shows and theaters and theme parks and cruise ships. So the devices I helped design ran the lights at Cirque du Soleil and Disney, and I got to go on site and see some of these things, which was really cool for me. Yeah, so that co-op job rolled into a real job after I graduated. So I moved to Calgary for that work and continued developing lighting entertainment equipment, and did that for about 4 or 5 years. And then I met my wife there in Calgary, and we decided we wanted to move back to Vancouver, where I grew up. And in that move, I also felt like I was interested in a change in career trajectory. I'd been a lifelong subscriber to Scientific American, and I'd been reading about, recently about the Human Genome Project, which at the time was new. And this was, as you all well know, the first time and that a human genome had been sequenced. And not only that, but it would be the first time ever in humanity that the first human genome will be sequenced. And I found that super just intriguing that we had this, and even once we'd sequenced it, we had this 3 billion nucleotides that we had virtually no idea what it did, this giant mystery to solve. It was the first time we were going to get to solve that mystery, and I thought I would like to be involved in that. So I, on the topic of networking, I had no biology experience, and I, any cocktail party I went to or whatever, I was just, I just say out loud, I think I want to work in cancer research. And then one day at choir rehearsal, I sing in choirs and have done for a long time. I mentioned this to somebody, and they're like, "Oh, you know what? I have a contact." He was a professor of microbiology at UBC, and he says, "You know what? I have a contact with someone at BC Cancer," and that rolled into a job interview where I convinced them that I am an excellent programmer that knows nothing about biology but has a passion for learning about it. And they hired me. In fact, not just they, but Enoch Birol hired me right here. And so he had a project and a plan and a problem that he wanted to solve, which is that Illumina short read sequencing had just come onto the market. And in fact, it was so new, it wasn't even Illumina at the time. It was Solexa sequencing by synthesis. And this was brand new technology. It was short read, high throughput sequencing. And Enoch wanted to assemble a human genome using this technology, which would make it massively cheaper than the Human Genome Project, which cost something like $1 billion for the first human genome. And so it was technically a very challenging task. It was something like 1,050,000,050 base pair reads. The reads were small, repetitive, fragmented, and from this we were going to try to assemble a human genome. And so we had a team of 3 of us, Enoch Birol supervising, Jared Simpson and myself, and we developed and wrote the software Abyss, which was the very first software tool to assemble a human genome de novo without a reference from short reads. And that's where I got my taste for genome assembly. And I was staff at the time at BC Cancer working as a programmer, and decided I wanted to continue this in a PhD. So at the time it was a master's. So I started the master's program in bioinformatics, did my year of coursework at UBC, on site at UBC, and then fast tracked into the PhD program and came back to BC Cancer to continue my research with Enoch. Jared Simpson had also left his position as staff. He had moved on to do his PhD, and his focus of research ended up being long read assembly, where, I was mentioning how short reads were, 50 base pairs. And at their peak of length, they only got up to 250 base pairs, just 5 times longer. We've all seen the chart of the Moore's law, massive exponential increase in throughput and decrease in cost of sequencing, but that just never happened for read length. It doubled and tripled and then just stopped and stagnated. Long read sequencing, on the other hand, just kept getting longer and longer. And so, and genome assembly a lot of it went in that direction. And in Enoch's lab, we continued to go in a different direction, which was to take all the benefits of short read sequencing and cost benefits of high throughput. And there was a young company at the time, there still is, at the time they were young, named 10x Genomics. And they had an instrument which could generate short reads that were all derived from the same large molecule. So you take a genome, you fragment that into large molecules. And then from each molecule, you get a bunch of short reads so that you know all the short reads came from that same large molecule. Large here is about like 100 kilobases. And so this gave you both the advantages of short read sequencing as well as the long range context of long read sequencing. And so with this, we applied it to we already had the experience of assembling large genomes. Human genomes were at the time the largest genome that had been assembled, but of course there's large genomes. And in collaboration with a group in the Michael Smith labs at UBC, we were looking at assembling even larger genomes. So conifer genomes like spruce, for example, are 20 gigabases, which is 7 times the size of the human genome. So these are still are, but at the time were amazingly complex to assemble. In fact, it hadn't been done yet. So we continued our field of research in assembling large genomes, and we did this with linked read sequencing, and we were one of the first teams to assemble a conifer genome from any technology, but particularly from linked reads, and did the spruce genome and the western red cedar genome in that fashion. And that was my PhD research. So continuing to develop bioinformatics software, but not only developing it, but applying it to collaborating with teams here at UBC to apply it to real projects, biological projects, that is. So that wrapped up my PhD, and during which I also had my 2 kids. They're very loud and they keep me busy and they're a lot of fun. So having wrapped up my PhD, I mentioned the technology we were using was 10x genomics linked reads. And through that and through grad school, I always paid a lot of attention to getting to network events like this and chatting with other fellow students and chatting with people in industry and making contacts, and also had the benefit of being able to take my research and present it at various conferences, mostly ISMB and Recomb. And in that got to meet people who worked at 10x. And they're a great group of scientists and established a bit of a relationship with them. So that once I had an expertise in their technology from using it during grad school, so that once I graduated, I moved from being a customer of theirs to getting hired and working for them as a research. So my role at 10, and that was about 6 years ago, and I've been there ever since, and I'm still there. So my role at 10x is in research and development and in computational biology. And 10x designs and manufactures instruments, kits of reagents, as well as we build the software that analyzes the data that it produces. So that's where I come in. I'm part of a team that develops the Cell Ranger and Space Ranger software, which 10x gives away for free. It's a value add to the instruments and kits that we sell, and analyzes the data that they produce. So I am nominally I'm a programmer, is supposed to be my job, but having been there for 6 years, the more you learn about the more institutional knowledge you have, the more use you are doing other things than programming. So, and I enjoy those other things. I still enjoy programming, but a lot of my day-to-day is communicating, troubleshooting, problem solving, facilitating inter-team communication. Because one of the things I love about 10x Genomics the most is the people and the interdisciplinary nature of it. Because at its core, it's a microfluidics company, right? So that means they employ physicists and engineers, and of course, it's a biology company, so you need biochemists and you need programmers, and that's just the R&D roles. And then on top of that, there's lawyers and marketers and everybody else. And I talk with all of those people I just named. As well as all the customer-facing roles, which are super important too. I am permanent remote in Vancouver. 10x is based in the East Bay of California. So my workmates here, I've only just hired my first R&D employee here in Vancouver, who just recently graduated from UBC. His name is Bara. And all my other all the other staff here in Vancouver are customer-facing roles, like sales, support, and field application scientists. So when you're looking at roles and jobs, you know, consider customer-facing roles too. Yeah, so yeah, like I was saying that a lot of what I do is just, well, not just, but is troubleshooting and prioritizing and not facilitating, making everything work. I keep the lights on sometimes is what I say, and I enjoy that work. Yeah, I guess I'll say a couple more things about my career trajectory within 10x Genomics. And that's that so it's project-based work, which is something I really enjoy. I work in new product design, which means, and that's what drew me to engineering. I like engineering is designing and building something, right? So you have a team assembled with various fields of expertise whose purpose is to gather all those expertises to make something that you couldn't make on your own. And I like that really driven, focused energy that you get by assembling a team. And we do that at 10x with, so we assemble from all the employees available to us, we assemble a team because we have multiple teams, each one building a different product. And we assemble a team we have all the expertise needed to build this new product. And then we do that. So the first one I was assigned to was taking our existing Visium spatial transcriptomics technology and adapting to that to FFPE slides. So FFPE is formalin fixed paraffin embedded tissue. It's a common archival format for tissue. And if you can analyze that FFPE samples, it vastly opens up the types of samples you can open to, for example, biobank samples, which are long-term archival storage. Because getting access to things like, for example, fresh biopsied tumors is complicated. You have to have relationships with doctors and hospitals, and you need cold chain management from when it's biopsied to your research lab. And that's all incredibly challenging logistics. So if you can analyze FFPE tissues, which are stable at much higher temperatures, then it simplifies that, makes research easier. So yeah, so the existing Visium technology required fresh frozen tissue slides, and we're going to adapt that to FFPE. And that sounds like it would be straightforward. Well, no, it doesn't mean, anyway, the reason it's challenging is what I was going to say, is that the formalin cross-links the RNA with surrounding tissues. That's what stabilizes it, but that makes it very inaccessible. Like the purpose is to stabilize it, right? That's what stops it from degrading. But for the same reason, it also makes it completely inaccessible to all the enzymes that you need to use standard sequencing techniques, which involve, for example, reverse transcriptase, which binds to your mRNA molecule and, has to process across the entire length of the molecule. If it hits a cross-link thing, it's going to get confused and fall off, and that makes it inaccessible to all the things you'd normally do. So we had to come up with completely new ways to interrogate and count the RNA to make this successful. So that was the first project I was involved in. And it was smashingly successful. And ever since then, I moved from their spatial technologies to their single cell technologies, which are, okay, sorry, stammering. Single cell technologies take each individual cell and put it into its own microreaction chamber using microfluidics so that you can construct a sequencing library for each individual cell and interrogate and count the contents of each cell by cell for millions of cells. We just launched a new product this week, in fact, one that I was working on. And with a single 10x chromium chip, you can now do single cell sequencing on 2.5 million fixed cells, and I've continued to work in the fixed RNA space. That's been kind of my specialty at 10x. So yeah, I think that brings me here to today and like this week's product launch. So that's been the trajectory of my career from Lego technician at the age of 3 to me now, jack of all trades and chatty person. So that's all I had to say to you all. Thank you for your attention. And And I'm happy to take questions now, or feel free to reach out on LinkedIn. And keep an eye open on job postings at 10X. And if any of them interest you, feel free to drop me a line and I'll make an introduction if I can. If you're interested in customer facing roles, I might be able to introduce you to one of those people here in Vancouver. And if you're interested in R&D positions, I obviously know computational biology the best, but I can try to talk a bit about other roles that you might be interested in. All right. Yes. Mm-hmm. Yeah, coming from, yeah. Yeah, so good question. The question was, how was it transitioning from an engineering background to learning about biology? And one reason I really like programming is because it's a skill, it's like a tool that you can apply to a vast number of fields, you know, from biology to finance to, and those are called the application domains. And you have to learn the application domain. Often as a programmer, you have to learn the application domain on the job if you haven't learned it in school. So for me, what that meant was when I went to a lab meeting, I didn't understand a single word they were saying. The only reason I went to the lab meetings was to sit there with my notebook and write down all the words that they said that I didn't understand. And then I spent the rest of the day Googling the words and learning biology. And specifically the narrow, laser-focused, narrow subset of biology that was relevant to what we were doing in the lab. So it was, so it was a good way to learn it. Like, just as a random example, they kept talking about back assembly. I had no idea what a back was. I was like, after the meeting, I was like, are we assembling like backs? And they're like, oh, no, bacterial artificial chromosome. Yeah. So that is what that's coming from engineering to biology. And it's really, just that, that's how I did it. So I just pay, I don't know if I want to say much more about that. It's just needing a desire to really want to learn and have the time and energy to do that on your own, which is also easier before you have kids. So coming the other way, it's a little different because you're coming with the domain knowledge and you're learning the skill, the tool, right? Which is a fundamentally different thing. And so it means that you need to learn that tool. Okay, so there are problem-based learning websites like Rosalind, for example, where they give you programming problems to solve. And it starts really easy and gets progressively more difficult. And you can do it with any programming language you want to learn. So that's another thing you have to do on your own, is learn the programming language. But having this framework of like problem-based learning helps me. And that's how I was learning new programming languages at the time, and also learning bioinformatics. So it's going to be a lot of like like time on your own to learn these things is what I think. You can learn them in school, but it's going to also take, like programming in school, but it's going to take as much time again outside of school to bring yourself to level. Yeah, but if you like you have to like programming. I'll say that. If you enjoy it and you want to do it on your own time, I absolutely trust that you will get there. Mm-hmm. Yeah. Yeah, I do a lot. I'm very tired and very busy, but I enjoy it. It's by choice. What do I do? Oh, so I'll talk about one nice advantage post, pandemic or panini is that, there's more remote work and more flexible work. So similar to the previous speaker, and sorry, I've forgotten your name. It was Venus. as long as you attend your, get to your meetings and get your work done, you can largely schedule your own time, which means that I can do things like take time off work to see my kids' school play, so it comes down to time management and having a good relationship with your manager and team, and being able to, and yeah, just balancing all the cards, throw all the balls in the air and hope you catch them. That's I don't know, but I still have I have a lot of fun. It's 10X has a good work-life culture as well, as long as you get your work done.
Have you ever considered that path, or were you like, I'm gonna go into it? And if so, what the pros and cons based on your perception?
Yeah. Okay, I won't be able to talk directly to being a prof because I never was. I'll probably defer to Ananch to answer that trajectory. What I did was during grad school, I tried to keep although I was always kind of inclined to go toward going back to industry, I tried to keep the door open to having a CV that would have led itself to an academic career, to publishing papers, presenting at conferences, so that I felt like I still had that avenue open to me, I think, when I graduated. I will say that my impression, and again, not having done it, is that you have to be you're running a small business when you're running a lab, right? So you have to be you're fundraising, you're hiring, you're managing, and you have to want to do those things. And I don't think I didn't want to do those things. So although, like I was saying earlier, most of my day is not programming now. Most of my day is, again, communication, and other things. No matter what job you get, it's going to be a mixture of responsibilities. Yeah, I'll leave that there. It will. Mhm.
So I'm wondering with ChatGPT now,
Mhm.
proposing to do with how important is learning about coding, and what is learning coding change how you think about things? Does it sort of make you sort of consider problem solving in more logical way? Now, what your advice over you to the students here who may not have the coding experience, should they be doing that if they seriously consider about it?
Yeah, I think coding is an incredibly useful tool to have in your belt, even if it's not what you plan to do day to day. And at the very least, it allows you to recognize problems that ought to be automated. If you find yourself doing some incredibly repetitive task that's going to take you a week, you can, to consider automating it or hiring somebody who can automate it. And being able to recognize those problems. Programming just naturally makes you have to be really rigorous about thinking about how, the steps to solve a problem, because you're telling a computer to do those steps for you, and a computer this might change with artificial intelligence, but currently has no way to intuit the things you don't tell it. And so having to go through that rigorous process is helpful to understand the problem. And yeah, I think developing some programming skills of your own is kind of like learning how to maintain your own car, in that, like, you might not do it every day, but learning that vocabulary and it allows you to talk to a mechanic with the vocabulary you need and understanding of the system, so that that communication goes more easily. So if you find you're going to be even just communicating with or managing programmers as part of your job, it's helpful to have some of that skill set in your pocket. Yeah. 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 Mhm. Yeah, actually, that's a great question, and I wish I'd mentioned it. So thank you for asking it. At 10x Genomics, the cell ranger is implemented pretty much entirely in Rust and Python, and it was originally Python only, and the heavy lifting is all done by Rust now, which is so you don't. Good news is you don't have to learn C++ anymore. It was a useful tool for its time. We have moved on. Rust it took a long, like I was always looking for a language that was as good as C++ and better in every way. And every language after C++ until Rust, was better in some ways, but not other ways, and didn't do everything that, it was always a compromise. And Rust is the first one where I can just say hands down, wholeheartedly, it's just better in every way. It's my favorite language right now. So it's so that's got my vote. And that's a compiled, strongly typed language, and it's good to have two languages in your belt, compiled, strongly typed language as well as a scripting language. And Python is probably the hands down winner for that. I've got some, I don't know, if I'm sitting down with you at a beer, I've got some opinions about Python. It's got a lot of nasty corners that that make me really frustrated with it. But it's still it's still just like everywhere. And it's a great tool to learn as well. Oh yeah, and also R. And my primary use for R is statistics and plotting, and mostly it's plotting for me. I do some some statistics and a lot of plotting. But for plotting and reporting, it's just hands down the winner for sure. And that's all like when I do data analysis at work, I have to communicate the results of that analysis to my audience, right? To managers and so learn data visualization, not just as like, barfing a scatter plot onto a page, but how to, what is the best way to present a figure that communicates the message you're trying to convey in a way that's as clear as possible to your reader. And I don't know, it's honest, it's like part of the funnest part of my day too, is it's the most artistic and aesthetic thing I do in my day is making a good interpretable plot. Yeah, so that's my plug for art. Yeah, no, I'm not talking. Yeah, performance-wise, they are very similar. I'm not the other. It's more that the C++ is just packed full of foot guns, ways that you can write programs that will just explode in subtle ways and not give you any hint of what went wrong. And Rust is the newest concept that it incorporates is what's called the borrow checker, and I won't. But it's basically a safety system that prevents it for so much. It's something that used to be said about Haskell, which is a language you're probably not familiar with. That is, if you can get your program to compile, chances are it's correct. I mean, that's smoothing over a lot of other ways you can create bugs, but Rust is similar in that way. And that's so coming back to Python, there's so many ways that Python can fail at runtime that should have been caught at compile time if it were compiled, but it's not compiled, which is why it only fails at runtime. And that can be super frustrating when you're, 12 hours into a run of a program that's assembling a genome or something and then explodes in the 12th hour due to like a type issue, right? And type issues, like I was saying, should be caught at compile time. So that's one of the reasons for my preference for typed languages and compiled languages. Oh, right, industry. I haven't been following that. I've got to feel, so my prediction is that will be the trajectory, is that the industry will move away from C++ and Rust, and you're seeing a lot more Rust in industry. There is a chart that's updated every year, and I wish I could give you more than that, but it tracks programming language popularity in industry, and it's like it's like the Rotten Tomatoes of programming languages or something, and you can see how the languages are changing over time, and how they're being uptaked and took up into industry. So that would be where I'd look for that. Oh, Inach says IEEE Spectrum maintains that. So that's where I would check to answer that question. All right, I'm probably well over time. So
Thank you very much.
Yeah.
Thank you very much. All right, everyone. Thank you so much, first of all, Dr. Jackman. We're just going to move on to a little bit of a social intermission, get up, stretch your legs. There's some snacks and drinks at the back. We'll be back in 10 minutes, and then we'll have Dr. King presenting their research. Thank you very much. I think so, yeah. Hi, everyone. We're just going to get started in just a moment. So if everyone could take a seat. Okay, everyone. I think we're ready to start. Without much more ado, here's Dr. King. They're presenting their research. You can introduce yourself.
Make sure this is the right height. Last person who spoke who is much taller than myself. All right. So it's very much an honor to be speaking to you today at this Van Bug. And I was asked to give a science talk. However, I did know that the people speaking before me would be giving career trajectories, and none of them were academics. So I've got a few slides of career stuff and happy to sort of answer any questions if academia is a career path that you'd like to pursue. And then I'll segue into the cool sciencey bit. Okay, so career in a nutshell, it's like a sandwich with 2 slices of bread, and both are called UBC. So I did my undergraduate degree in biology at UBC, and then just recently arrived as professor in Canada Excellence Research Chair, the departments of zoology and microbiology at UBC. 20 years later, I did have a life in between. Okay, so during my bachelor's degree, I learned a lot about evolutionary biology from Dolph Schluter. And worked a lot with sticklebacks. There's a lovely picture of sticklebacks here. But I had also learned a lot about parasitology from some professors I had at UBC and became really interested at that point in infectious disease, but from a wildlife perspective, not a human health perspective. And there was one other group in Canada at the time that was looking at parasites and infection from an ecological or evolutionary bent, and that was at Concordia University in Quebec, Montreal, in Quebec. And so I moved to do my master's degree there, and then there I was very much an applied ecologist. So I had collected sticklebacks in the fields in British Columbia and in these lovely glacial lakes. And here I was in wetlands collecting frogs, and we were looking at the impact of agriculture on frog disease. But then after that point, I decided to go back to my first love, evolutionary biology, and got in contact with a professor at Indiana University who was looking at host-parasite interactions from a more theoretical perspective and looking at how coevolution between hosts and their parasites could maintain genetic diversity in the host and the parasite populations. Something called the Red Queen hypothesis, which I can elaborate on if anybody's interested. And that involved going to New Zealand every year for for several years, every year of my PhD, and working with the snail parasite system. So lots of traveling about. So then I got my PhD, and I'm happy to talk to you about the reasons I continued on in academia. But I wanted to go to the UK, which was a hotspot for infectious disease evolution. And was very lucky at acquiring a Royal Society Newton Fellowship as a postdoctoral researcher to work with Greg Hurst at the University of Liverpool. And there I switched systems again. I started to work with C. elegans. Do you guys know what C. elegans is? It's like this workhorse lab model, right? That you can work with in the lab. Its genome is, it was one of the first animals whose genome was fully sequenced. It has these lovely genomic toolkits that are associated with it. And I also started to work with Nisonia vitripennis, which also has lots of genomic tools associated with it. So I was a field biologist, basically, beforehand. But very quickly, when I started my postdoctoral fellowship, I realized that particularly to work with these model systems, that genomics and bioinformatics was going to be the way forward and allow us to really look at evolutionary patterns, not just in terms of phenotypes, which was mentioned before, but in terms of the genotypes and being able to connect these two things together. And so I started to learn some bioinformatics and some sequencing procedures. And then this is where I admit to you that I'm a massive poser in this audience, because after this, in 2013, so what's that, like 11 years ago, I then went on to an academic position where my practical knowledge for bioinformatics stopped. And I was now in a position to apply for grants to hire people who had this bioinformatics skill set. Or if they didn't but wanted to, I knew of collaborators they could work with to get that practical knowledge or courses I could send them on. So I became an avid reader and communicator with people engaging in bioinformatics techniques, because I knew about how, and I knew what it could tell us, but I couldn't do it anymore. So when I talk about the sciencey bit, if you have any questions about the practicalities, I might refer you to the paper or the clever postdoc that did the work. But nonetheless, at least you get to see a different perspective on how bioinformatics training ensues, and then also what it can elevate you to in different ways. So after my postdoc, I jumped to an associate professor position at the University of Oxford, where I then remained for 10 years and built my group. And it's only recently, as of last year, that I was recruited back to UBC, which I jumped at the chance. It was a nice opportunity to come home after being abroad for 20 years, and be able to take some of the knowledge of the systems that I've been using in the past, and the systems and the technologies we've been able to develop at Oxford, and really bring them together. Oh, and the one last thing I was going to say about this is that I'm showing all these different animals for a reason. I'm referring to my previous experience as a field biologist for a reason. The work I'm going to tell you about today is very lab-based and genomics-y, right? But all of the work we do is inspired by natural field patterns. And we use our experiments and our genomics in the lab to infer potential processes that could be leading to those patterns in the field. And I think that will come through in how I do the sciencey bit. Okay. So, oh, you can't see things. A lot of my talk you can't see. No, it's just a piece of PowerPoint. I could give a chalk talk, but I don't think you want that. Do you want me to get my laptop? I guess while they're doing that, does anybody have any questions about academic route or jumping from country to country or doing work in different places or anything like that? Help me waste time. Go for it. Yeah, so that's another reason why I brought that up, is there are different kinds of scientists, I've realized. So ones that are very system focused, and that allows them to drill deep down and do really, really excellent science that way. For me, I've realized over time that I'm sort of more interested in sort of the general patterns that exist across systems, which isn't uncommon for an evolutionary biologist. We're looking for evidence, and we're investigating potential sources, modes of selection, evolutionary patterns that would be relevant regardless of the organism. And also I think I just get a bit bored of the system and I want to ask questions that I can't address using that particular system. So I've always tried to frame it as a positive. And whenever anybody has a critique, which is fair because it's hard to learn a new system each time. But I'm always sort of armed with this knowledge that we're investigating patterns that are broad spectrum. And regardless of the organism, it's going to be relevant. Whereas potentially, if you're looking at patterns that are more specific to a particular organism, it doesn't go beyond that, depending upon the question you're asking. It's never come up, but I've always been ready for it, that critique. Go for it. Yeah. Oh, yeah. So that's a good question. I really enjoy grant writing when I'm not looking at the end of my previous grant. So when I've got a bit of time before, I've got a bit of a buffer zone, grant writing is great in being able in helping, I guess, frame your thoughts. The downside is when the grant obviously gets rejected, and sometimes there are very few places that you can send that otherwise, unlike a paper, unlike a publication. Canada has a very, very different funding model, particularly through the NSERC Discovery Grant system, where the funding rate's about 60%. Mind you, you don't get a high amount of money, but you get enough where you can sleep at night. It's enough to sort of fund a PhD student, more or less. If you want to apply for more money, there's CIHR, obviously, there's Genome BC, and there are other sort of NERC programs as well. In the UK, we have a UK Research Council, and the funding rates are really low. You get a large amount of money, but half of that goes for overheads to the university. So my last grant over there was an £800,000 grant, and £400,000 went straight off into the Bodleian Library and into the department to keep the lights on. So here, that's not the case. So you get to keep most of what you get, depending upon the source. Yeah. What are you doing? So right now? Fab. Oh, thank you for that. Thank you for the question. Yeah, and actually, if anybody's interested in going to Europe at some point for graduate training or postdoctoral training, UK, I've also got experience on the continent. I'd be happy to chat about different ways you can do that. There's way more funding for postdoctoral researchers, for example. Okay, excellent. You can see my continuum. Right, so virtually all plants and animals, much like all of you out there and people in the virtual lecture theater, are host to numerous microbes. You know this, but this realization has fueled unprecedented interest in the multifarious impacts of those microbes on host biology. These microbes need not merely be strictly passengers. They can be harmful, of course, but they might also be helpful. In fact, the symbiotic interactions between hosts and their microbes can vary along what we call a parasite mutualist continuum. On the extreme parasitism end, we have an example of a Plasmodium parasite blowing up the red blood cells of their bird host, but obviously Plasmodium can also be in mammals, obviously causing a great deal of harm. Nobody's ever said that they benefited from malaria infection. And then on the other end of the spectrum, we have an example of the hundreds of millions of years long, deeply interdependent relationship between some aphid species and their inherited intracellular symbionts. These relationships are largely nutritional in nature, and you can't tease these two things apart. The symbiont nor the aphid can exist without the other. However, most interactions between host and their symbiont, I'd venture to say, are movable along this continuum, either in ecological or evolutionary time. Small jumps, but also big jumps. So an example of a huge evolutionary shift from a mutualistic host microbe interaction to a parasitic one is that of the widespread plant pathogen Pseudomonas syringae, which evolved from mutualistic ancestors via horizontal gene transfer of type 3 secretion systems, essentially toxin production. An example of an extreme shift in the other direction, there are these entomopathogens in cicadas, which now take over the metabolic role, which was originally done by degraded endosymbionts. So these are massive shifts, but again, I talk about these little shifts. What about parasites becoming more harmful or mutualists becoming less helpful? So one research line that my group has been pursuing is that of looking at the drivers and consequences of these shifts along the parasite mutualist continuum. And we view these transitions through what are called protective symbioses. Now, what are they? So increasingly, evidence is mounting suggesting that the microbes colonizing you and a whole bunch of other organisms in nature are strong determinants of your susceptibility to infecting pathogens, parasites, and parasitoids, not for humans, but for insects. And in fact, microbe-mediated protection seems to be a widespread phenomenon observed across animal species and plant species and also in humans. And so, for example, you've heard of probiotics, right? Potential probiotics. So in theory, these are microbes that can protect you from infection. These are some different systems which have highlighted how protective microbes can operate. So you can have resident microbes that upregulate the host immune response, and so any pathogen trying to come in now is meeting an already charged host ready to fight it. So that's a host immune-mediated pathway. You have examples of also direct combative interactions between protective microbes and pathogens. So you can have just strictly resource competition, and that's been observed in Drosophila. So, and also in your gut as well. So you have microbes that are already adapted to exploit your resources. And again, any pathogen trying to come in is going to be an inferior competitor. However, we've found that across the tree of life, where these mechanisms have been investigated, that it's actually toxin-mediated production that's the most common. Commonly observed, I should say. We don't know if it's the most common. And that's when these protective microbes will produce some sort of toxin or antimicrobial, which directly kills pathogen cells. And we've shown this to exist in a C. elegans system, which I'll go into a bit more in a few slides. So there are all these different mechanisms. And so these protective microbes, these protective symbionts, have huge environmental importance, potentially shaping patterns of infectious disease out in nature. But because they're so important and because they're so effective in many other circumstances, they're also attractive candidates for disease control, particularly in an era of unprecedented resistance to antibiotics. And so here are two examples of where microbes are being applied in a protective capacity. Here on the left is a picture designed to represent fecal transplants. If you don't know what that is, I don't recommend looking it up on Google. Image. I've given you a very sanitized image here. where you essentially have the microbes from healthy individuals transplanted into the guts of patients suffering from debilitating infections. It's proven to be quite effective against C. difficile infections, for example, hospital-acquired. On the right, there have been a flurry of new studies over the past, say, 5 years, but 10 years previous as well, piloting ways to introduce a bacteria called Wolbachia into novel insect hosts, particularly mosquito vectors. And this Wolbachia in these hosts is very good at preventing the mosquitoes from acquiring dengue. And then a few studies ago, or a few years ago, a study showed that then releasing these Wolbachia infected mosquitoes into the field actually then reduces the transmission of dengue to humans. But that's quite cool. So these microbes are of huge importance. But as somebody who's interested in parasites, and if anybody here studies infectious disease, and the question is, now what? So pathogens and parasites are bloody good at evolving in response to barriers to their establishment. Have a look at AMR, right, antimicrobial resistance, or how COVID evolved in response to vaccination. Okay, so what are the potential evolutionary consequences of using a diversity of protective microbes? How might pathogens evolve in response? So the question is, how do protective symbioses shape pathogen evolution along this continuum? At the introduction, I highlighted these massive evolutionary shifts. Now we're sort of talking about wee ones along this spectrum. So you have a pathogen that's here. Does it move to become more benign, or does it become a lot more harmful or more resistant to the action of protectors? So we use lab-based systems and a process called experimental evolution with follow-on bioinformatics to be able to test these kinds of questions in the lab and determine what kind of evolutionary paths might these pathogens follow. But then there are some follow-on implications, right? Because can we now forecast how pathogens might evolve in response to these protectors in more applied situations? So today I'll be talking about two separate studies and two separate systems where we've answered this general question. So the first, we're looking at the evolution of pathogen virulence with the C. elegans system. So if there's anybody out there who knows about these worms, one of the great reasons for why it's a lab workhorse is it has a lifespan of about 4 days at 20 degrees from egg to egg. We can easily propagate populations quickly. We can freeze them and keep them for later if we want to go on holiday or we're sick. From a host pathogen perspective, we can plate them on a petri dish with a bunch of microbes. The microbes can be ingested by the worms, and then the microbes can then infect the worms. So it's a little bit of a microcosm for how a bacterial infection might operate, for example. And so we can feed these worms protectors and pathogens, and all these microbes form stable population sizes in the worm gut, and they evolve. The other system I'll tell you about a little bit later, where we look at the evolution of pathogen resistance to protection, is with the bacterial pathogen and phages, which evolve super quickly in the lab. Like you can start an experiment and then come in a few hours later and you see evidence of evolutionary change. Super cool. So we were really interested in thinking about how pathogens might evolve in terms of their virulence in response to protectors. Just a bit of a plug from a previous paper that we published looking at how when the protector, in this case Enterococcus faecalis, produces toxins to kill Staph aureus, the staph evolves in a way to respond to these toxins, but it comes at a cost of its growth rate. So it grows a bit more slowly, staph does, in response to the protector. Grows a bit more slowly, it's less virulent, okay? But we then became really interested in this host immunity process. And could the evolutionary trajectory of virulence be different if the immune system is responding? So I'm an empiricist, evolutionary biologist. We largely rely on theory to test. We need theory, we need some maths. I work with theoreticians as well as bioinformaticians to be able to develop this theory. we didn't have it. So we borrowed theory and data on a concept called incomplete immunity. And a lot of this work was done by Dana Hawley's group at Georgia Tech. And there they work with a US house finch, which there are populations of these house finches which are being decimated by an emerging mycoplasma infection. Little infection that gets in the eye. Well, that's where it starts. And then it ravages the bird. Now, ideally, if you're exposed to an infection and you clear it, you have total immunity, right? You can't be infected again. That's kind of like the null scenario, the null hypothesis. And so here we have all these birds. There are two different kinds of pathogen. We have less virulent pathogen.
Testing.
Alrighty, you can hear me all right? Fab, okay. So where was I? Yes, so the birds acquire the less virulent infection, the immune system gets primed, the infection is again in the environment, but they don't get infected, right? Okay, so same thing here, but now we have a more virulent pathogen. And let's say this birdie here was infected by the less virulent one. Now it's an environment where this more virulent one comes in and it gets hit and it gets killed off. Okay, so in this case, the hosts are infected by the less virulent pathogen are not equally protected. Now let's say that the immunity is now incomplete. So the parasite, the pathogens can still get in, but the host's kind of okay with it. It's tolerant. It can deal with the infection. Okay. So we have an initial infection of the less virulent parasite, immune system's primed, hosts get reinfected, but they're okay, they're dealing with it. Okay. Same thing here, where we have the more virulent pathogen, it upregulates a stronger immune response, and now they can carry this hugely virulent infection. They're not bothered by it, but they're spreading that virulent infection about. So if that virulent infection happens to go into a host that hasn't had its immune system primed from a previous infection, it's toast, right? It's not in a good way. And you would get that response equally if they had been initially infected by a less virulent one. So what happens here is you get some protection against the more virulent strains, and hosts don't die from the more virulent pathogens, and they can live to transmit. Over time, what this does in simulations is it purges the pathogen population of the less virulent strains and selects for the strongly virulent ones. Okay, so this system and the maths associated with it, implying that this kind of incomplete immunity can actually lead to the evolution of more virulent infections. And so we borrowed this logic to apply to what happens when you have a protective symbiont that induces a strong immune response, and now you let pathogens evolve under this scenario. Would you get the same purging of less virulent strains and end up with a more virulent pathogen? Because the hosts are protected, but the pathogens can still infect the hosts, and the hosts are live to spread this more virulent pathogen. So this was a question of interest from an NSF postdoc of mine, Kim Huang, who's now independent fellow at Emory University. And she came to Oxford, she came to Oxford in the first week of March 2020 to start her 3-year odyssey in Europe. So we managed to take her to the pub to celebrate her arrival. And then we were locked down. And the UK locked down super hard, super hard. She's a trooper, super hard worker, and was really interested in the question. And so she wanted to know whether the symbiont immune priming could similarly select for higher virulence, just like previous mutation would. And for that, she used the C. elegans host. Can't really see that very well because I think the light's on. But this is a protective microbe found in soil that can naturally colonize C. elegans, Pseudomonas mendocina. And the pathogen that we used was Pseudomonas aeruginosa. I'm sure many of you have heard of it if you are interested in bacterial genomics. It's a widespread opportunistic animal pathogen. Infects humans in CF if they're suffering from cystic fibrosis, but it's also in the soil, it's in water, it infects a lot of different kinds of animals. Here we have a microscopic image of worms. This is the worm gut here. This is this P. mendocina, that protector microbe. It gets inside the nematode gut, upregulates the host immune response, and then it's cleared. Okay. And this was shown from colleagues previously at Berkeley. So Kim tried to repeat that, and she was able to. So here you have host survival on the y-axis. These are different kinds of nematode hosts on the x. So this is the wild type N2, if anybody knows worms. It's the canonical lab strain. Here's an immunocompromised worm. This is the immune pathway that's upregulated when colonized by this protective symbiont. We knocked it out to control for this immune pathway being present at all. And this is where worms had the symbiont or didn't have it. And what we see when we look at host survival in the presence of infection is that survival is much higher when worms have this protective microbe. It's doing its job, right? It's lower when the protector is absent. And then these worms are just rubbish. They're just dying. They don't have any immune response. And this immune priming, it's an incomplete response. It's different. You don't get total protection. It's incomplete. So what we see is when we look at the number of colony-forming units per worm, which is a way of kind of counting live bacterial cells on a plate, we find that even though the symbiont is present, the protective symbiont is present, there's still pathogens in there, okay? They accumulate less, so there's a difference between when the symbiont is absent versus present, but they're still there. Okay, so then Kim set up what we call an evolution experiment. This is where we do evolution in the lab. It's sort of a heuristic tool that operates in this space between mathematical theory and what happens in the real world. We're seeing what's possible from an evolutionary perspective. We decide who's successful. We decide what goes on over time. So she set up 2 treatments where she just had pathogens. She passaged successful pathogens from plate to plate of worms. Okay, so she took pathogens that successfully infected worms, decided those were the successful ones, and propagated them to the next generation of worms. But she also had a treatment here where the worms were colonized first with the protector. So like you have your microbes in your gut right now, right? And then you get infected later on. Okay, so she simulated that here. In both cases, it was just the pathogens that were passaged inside wild type hosts that had an immune response and ones that didn't. And this experiment went on for 14 passages and was replicated 5 times. So took about 2.5 months. Shockingly, she found that the results backed up our hypothesis. It doesn't happen all the time. In fact, it happens not so often. So this is great. So she found that as we predicted, as fit with the house finches and as fit with the math theory, symbiont immune priming seemed to favor the highest levels of virulence. So we took those pathogens that she'd evolved and she put them back into worms that were just sort of standard wild type, no protector in the middle. And that's how we measured the virulence. And we look at host mortality. And what we found is the highest host mortality was caused by pathogens that had evolved in a host with an immune response and that had the symbiont there. Okay. And then we see lower levels of virulence, not dissimilar from a no host treatment where we just passage the pathogen on a plate and the ancestor. So this was the starting point. And so these things evolve to become more harmful over time. So now for the bioinformatics bit. Okay, so we sequenced whole populations, but also several clones of each of our evolved lines at multiple time points. But I'm only going to show you data for a bit of it. So I'm going to show you data from the population where we had the highest levels of virulence emerge, and those where we had the lowest levels of virulence emerge, where we had an immunocompromised host. Okay, one of the speakers previously showed phylogenetic trees. Okay, so we can take the genomic data from our evolved populations. In this case, it's clones. So we sampled randomly 3 clones from each population. And then we had a look at what's called genetic distance. So how many mutations were accrued? And because all of our samples stopped at the same point in time, if a population has more mutations than another, it evolved faster. Okay, and so what we see here is these kind of bluey green dots represent the treatment where we saw high virulence evolve in wild type hosts with the symbiont. And these kind of purpley ones are where we had the immunocompromised host, no symbiont. And what we see on average is that the wild type worms, despite seeing a big change in virulence, actually didn't move that far genetically, which suggests that any mutations that gave them that virulence jump were sort of few. They're what we call large effect mutations. And these are the data that take the sum of the genetic distance across all of this treatment and all of that treatment to the ancestor and compare. And what we see is that the pathogens that actually evolved in the immunocompromised host traveled much further. They evolved much more rapidly than in the treatment where we imposed really strong selection. This is very, very interesting. I'm happy to talk about it more. It's also been found previously in studies which have looked at pathogen evolution in immunocompromised hosts, that these pathogens end up acquiring a lot more genetic diversity and evolving in different kinds of ways. The other thing we can do with this kind of data, so now this is population divergence. So this is based on clonal data. This is from sequencing the population. We can calculate something called FST, which is a measure of population divergence. And so we calculated pairwise FST between replicates within a treatment, okay? So if we have low FST, that means all the replicates in a treatment are moving in the same way. Very predictable, okay? If we have super high FST, that means we have lots of divergence between the replicates. And so one's going that way, one's going this way. And what we find here is just a wee little result, is that the worms where we saw the high virulence jump, we see the lower levels of FST, which suggests that we see more predictable pathogen molecular evolution, and we see more population divergence when the worms just have genetic-based immune response. We can also look at temporal changes across time, because we can freeze everything and we can resurrect everything from the freezer and then sequence at multiple time points. So we can sequence and explore evolution in real time. And so we looked at as for here, the number of loci that were where we had a significant FST value. And we see here that on average that there isn't a lot of difference in the trajectories between the treatment midpoints, endpoints, or the ancestor. But what it does look like is at the end, they all come out the same. So very, very similar FSTs at the end when you compare the ancestor versus the end of the evolution experiment. And this suggests that the forces shaping molecular evolution at the beginning become much less effective over time as the pathogen is adapting to the host. We also had some data looking at the genotype-phenotype link with virulence. I'm happy to share that if you're interested in as well. It's less conclusive. It turns out virulence is actually really complicated and polygenic, but happy to talk about that as well. So I hope that this kind of showcased the different kinds of molecular approaches you can take with evolution experiments when you're trying to predict how pathogens might evolve under different scenarios. So this little snippet, we've shown that in response to protectors, that pathogens can evolve to be quite virulent indeed. And we also saw lower virulence evolve in immunocompromised hosts. And that the evolution in these primed hosts was more limited. So we're looking at large effect mutations, and we see more parallel molecular evolution. There's only so many ways they can evolve. But we see accelerated evolution in immunocompromised hosts and evolution that's more diversified. So it might even actually be harder to predict if you're trying to do any kind of surveillance, for example. Okay, so that was a little talk about virulence and harm. What about resistance? Okay, so pathogens evolving to resist these protectors. This is particularly relevant in the context of therapeutics, right? You're trying to apply a microbe to treat a patient, for example, or in agriculture, you're trying to apply microbes to treat crops. But how quickly will those pathogens evolve just to resist what you're doing? So this was a question of interest in my group as well. And we decided to pursue it from the context of phages. So phages are really, really interesting. They're little viruses of bacteria, and they inject their DNA into bacterial cytoplasm, replicate, make little phage babies, and then blow it up. And then these phage babies kind of go out into the environment to look for new hosts. They're being developed in many places around the world as a possible therapeutic and an alternative to antibiotics, particularly as a last resort therapeutic when all antibiotics have been tried. There are several studies now published in quite high impact journals where these phages are quite effective. And they exist obviously naturally in your microbiomes. When they're applied in a therapeutic perspective, they're applied in terms of a huge diversity of taxa are applied. So different kinds of phages that infect the bacterial pathogen in a variety of different ways, a bunch of different receptors are targeted. So for an evolutionary biologist, this rings some alarm bells, okay? Because what you're doing is you're attacking the bacteria more, and you're also encouraging the evolution of a variety of different resistance mechanisms, potentially a generalized resistance mechanism. And so we were interested as to whether these phage protectors might, when applied as a cocktail, do more harm than good. This is a question of interest to a DPhil student in my group, Alex Betts, who went on to a junior research fellowship at St. Hilda's College. He now works in the civil service. So he now works in government. He was involved in the COVID response of the UK government. So in this case, our host isn't a worm. Well, it is, I guess it is a bit of a worm, but a worm without an immune response and doesn't move. So it's a test tube. But within this test tube, we obviously had media, and we had a diversity of bacteriophages, which Alex had collected from some of time he spent in southern France. So he went around the central wells and water fountains in Montpellier and isolated different phages. He also went around Oxford and had a dive in the Thames and the Cherwell. Don't recommend swimming in those. Don't recommend it. Saw lots of salmonella phages there. But he managed to uncover lots of environmental bacteriophages which could go after Pseudomonas aeruginosa. I should also say that Alex wasn't being strange in doing this. This is actually how a lot of phages that are end up being used in phage therapy are found. They're just found outside. So he assembled a bacteriophage community, and these are their names if you're interested, which went after different targets in the bacterial cell surface receptors, because the idea was to attack this Pseudomonas with a variety of different weapons. He then assembled in these test tubes these protector phages at different levels of diversity. So he had obviously one where no phages were present. It was just the bacterial pathogen happily evolving. And then you have one phage. So each of the 5 phage was just put with the bacteria on its own in every 3 phage scenario and in every 5 phage, or and then in the 1 5 phage scenario. So every 24 hours, a dilution was taken and then passage to fresh media. This experiment took 10 days. And it was published in Science. So Alex walked around with a target on his back for a while with a 10-day experiment. Okay, so what we found is that very, very quickly, the bacterial pathogen populations began to recover. So this is where you, this is the density of phages in the environment. It's all the phages together, not particular phages, but all of them within the treatments. And we see that this is happily bumbling along. Okay, it doesn't really change much on average. This line here is where you have no phages in the bacteria. So this is the bacterial population. No phages, so just happily carrying along. Okay, these are where the phages hit. Okay, and by the second passage, there's a nice order here where they're hit hardest where you have the highest level of protector phage diversity. Okay, so it's like is what's wanted in the therapeutic setting. This phage cocktail is hitting these bacterial pathogens harder than if one phage was applied. But by the third passage, bumps up. No impact of diversity at all. Okay, but it bounces back up. So this implies bacterial resistance evolution. Okay, they're evolving to resist the phages that are in that environment. So because we can freeze everything, we just take everything out of the freezer and we can look at what happens across evolutionary time. And we can do mixes and matches between different generations. Super cool. And so we can, these bacterial pathogens from what we call the future, it's the end of the experiment, basically, from the present, the middle of the experiment, and the past here, the past. And that would be essentially the beginning of the experiment. And then we do all combinations. And what we see is that across time, the bacteria evolved increasing levels of resistance and higher levels of resistance where they were hit with the highest protector phage diversity. So this phage diversity selected for stronger, higher resistance. Now the bioinformatics bit. So we sequenced all of our evolved populations. Pseudomonas aeruginosa has obviously a well-annotated genome. And so we knew exactly, this is why I got in science, so we knew exactly what the targets were for each of the phage. And so we could follow the evolution in each one of those genes that coded for the bacterial resistance. And so we calculated FST again. Again, that's a measure of how different an evolved population is per SNP from the ancestor. And we found that at the highest level of diversity, we had the most number of SNPs with a high level of FST, so most genomic change. And a lot of this evolutionary change happened in these known phage targets, such that of the 26 alleles that swept to fixation, 23 were also in a general phage target called LPS biosynthesis. Okay, so essentially the bacteria coats itself in like a bit of a slime and repels these phages. That was our fear, right? Sort of this generalized resistance. So I'll come back to that. And also, again, using phylogenetic trees, we can have a look at how much something has evolved from the same period of time across these different lineages. And so here's the ancestor here. And when we've sequenced the evolved populations, we see there's like a cluster of the low phage diversity ones here. But on average, we see most of the high ones here. There is a significant difference. So the high phage diversity treatment accelerated pathogen molecular evolution. It selected for a super high level of virulence. This is for any sort of evolutionary biology wonks out there. We can also use genomic data and bioinformatic data to have a look at the mode of selection. So there's, is that okay? You all right? Okay. So we can have a look at two different kinds of selection. Selection that kind of goes up and down, or directional selection, where a mutation arises and it sweeps to fixation. So in this case, where we have up and down of, say, like a pathogen resistance allele going up, and then the phage evolves a way to get at that pathogen no matter what. But then the more common it is, the more it knocks down that pathogen resistance allele, and there has to be a new pathogen resistance allele that would pop up for evolution to continue. This is what we call the Red Queen scenario, where we have fluctuating selection. Happy to go into that, has its origins in Alice in Wonderland. And this is the arms race scenario. And I know you've heard of arms races before. So it implies directional selection between two antagonists, in this case, the Pseudomonas aeruginosa pathogen and the phage. So we would have directional selection always going up, and here we'd see a change in the direction all the time. And so the point with the Red Queen scenario is that the more common you are, the worse you actually do. So the more common your immune system is, the more pathogens can adapt to you and knock you back. If you have a rare immune response, you have a rare defense strategy, the phages don't notice you very much at all. So you're at an advantage. And so we can use the genomics data and our information about what alleles are occurring in these defense genes to have a look at whether we have an arms race scenario or a Red Queen scenario. So we have a look at the midpoint of the evolution experiment, and we see that if this allele is really common, it's more likely to go down in frequency. It's common, so it's actually bad, because that means the phages get onto it and knock it back. And so what we see if we look at all of the alleles that were under selection in defense genes, we see that at the high phage diversity treatment, we see more arms race going on. So we can use the genomics data to look at the mode of selection, the speed by which it's operating, and how strong it is. Now, again, going back to this generalized resistance strategy, which is what we were concerned about. We wanted to know how general it was. So we assembled more phages from the environment. Different kinds of sources made all sorts of cocktails. And what we found is that the bacteria we evolved in the lab happened to be resistant against whatever we threw at it, whether they had seen it across evolutionary time or not. And so this is the bacterial passage in our experiment. And we can see that by passage 10, the high phage diversity treatment, these bacterial pathogens were the most resistant to whatever we threw at it. So imagine this could be an issue for chronic infections that you're trying to treat longitudinally with phage therapy. It's possible that the bacterial pathogen will just become better and better and better at resisting whatever you can throw at it over time. I don't know that that's true, but that's an example of a potential implication you could get from a heuristic experiment like this. So in the second story, I talked about pathogen resistance evolution. And indeed, if you evolve a very resistant pathogen that can overcome any protector you throw at it, I would call that a shift towards a more dangerous pathogen along that continuum. You've seen here that a diversity of protector phages versus the monocultures impose strong directional selection for super high resistance. And we saw really accelerated sweeping of these generalized resistance mutations, LPS biosynthesis, and that ended up coding for resistance to novel phage protectors, whatever we threw at it. Thank you very much. You guys are probably tired and hungry. Go for it. Yeah.
Are future antibiotics still going to be small molecules, or is there any future where they would be phages in a way that's not plastic?
Yeah, I don't know. So I know that work is ongoing looking at the combination of it. I don't think anybody wants to get rid of antibiotics or replace them entirely. But I know that there's clinical work going on looking at the combination of antibiotics and phages together. Nice thing about phages is they can evolve to overcome any pathogen resistance that's emerging. And so people are trying to be able to identify phages that work together with antibiotics. Perhaps there's a trade-off between resistance mechanisms, right? And if they want to resist the phage, then they can't resist antibiotics. Did that answer your question? Go for it. So I know that people are also looking at sequentially adding phages as monocultures. So rather than putting a whole diversity on at once, it apply that diversity, but in stages. Perhaps such that if the bacteria evolved resistance to the first one, it can't now evolve resistance to the second one you're throwing at it. So again, playing around with those constraints. And also, if you're going to use monocultures, I have colleagues who are also looking at being able to evolutionarily train those phages on bacteria that are already resistant. So then if they're ready to go and ready to fight resistant bacteria, then once they're applied to a patient, then they have even more of an edge in treatment. I'm not doing that work though. Go for it. Okay. I'll take a stab at it and answer your question. If I don't, we can chat over a slice of pizza. So you're talking about, why should pathogens evolve to become more virulent? Is that right? That it's already reached its max, it's already super harmful. Why would it evolve to become more harmful? Is that what it is, or? Because the selection pressure is always there favoring it. So essentially, the idea is that so any sort of adaptive trait is going to be a benefit and a cost. And the benefit to being highly virulent is you're actually extracting host resources, you're growing, right? But you don't want to be so virulent that you kill your host prematurely and you can't transmit. If you're in a host that's able to tolerate you, you can be super harmful in theory, and you won't kill the host. So you're not paying that cost. You're not killing the host prematurely. The host is doing just fine. Thank you very much. So that means that once you then get out into the environment, encounter hosts that can't tolerate you as well, you're now super, super harmful. I probably didn't answer your question, did it? Is that all right? Oh, that's all right. We'll keep having a chat. Yeah. So the nice thing about phages is they're super host specific. You can get phages that are even specialized on particular genotypes. And so that is, I think, considered less of an issue with phages as opposed to more general antibiotic application. But there's also great strength in antibiotics being general, right? You don't have to know what the bacteria is to treat somebody. You can just go for it. Whereas with phages, because they're so host specific, you would need to know, you need to culture it. So there's a bit of a delay there. So that's certainly a cost to that specialism. Yeah. Oh, that's a great question. So we didn't look at horizontal gene transfer in the bacteria. We looked at it in the phages. And we see the outcome of horizontal gene transfer. So like the mixing of DNA, right? So we saw hybrid phages being produced in these infected bacteria, which then leads to another question. If you have these hybrid phages emerging, are they then really good at going after everything? Or are they kind of hopeful monsters? They're just kind of rubbish, and then they fall out of the system. We did look, I don't know if this answers your question, but we did look at costs, and the bacteria that did evolve to resist the phage cocktail grew more slowly. So resistance was effective, but they grew more slowly and probably on their own would be less good at exploiting a host, which would be a massive benefit. But we didn't introduce, like, for example, antimicrobial resistance genes into the mix. And that certainly would have been a good example of seeing how they would have flourished under that environment, particularly because phages can move AMR plasmids and genes about. So thank you. That's actually really good. Yeah, cheers. It's a good idea. Might bug you about that some more. Yeah.
Thanks for the exciting talk. When you are applying those local monocultures, how about cycling? So always in monoculture.
Yeah. Yeah. So I apologize. That's what I meant when I said sequential exposure. Yeah. So people, yeah. So what do you mean by cycling? Yes, yeah. So that's what I mean. So that's the same thing, yeah. So you take, phage 1, you expose the bacteria for, a day, and then you apply phage 2. So you're still applying diversity across time, but one by one. And people are looking how that can be exploited as a way to use the benefit of the cocktail, which is a diversity of weapons, but in a way where you don't get the evolution of this generalized resistance. Yeah. I want pizza too.
It's wondering, like, how you verify that when you say evolution, these two new mutations that are introduced in the population after they got in the phase without any type of selection pressure, or are these in the existing pool that just get amplified?
No, I should have mentioned. So all our evolution experiments start with a single clone. So it means, yeah, no, absolutely. I mean, everything starts with the product of that single clone. I mean, quite right. If you pick a clone, you grow it up in vitro culture, it's going to have acquired something. But then that mixture seeds everything at the same time. And then all the lines are started from that. Absolutely, absolutely. And we also focused on these phage targets. We didn't use any long read sequencing in this, so we used a short read, but then we conducted follow-up Sanger sequencing on these particular gene regions that we were interested in, particularly the ones coding for LPS. Is that answer your question? Yeah, yeah. Well, we used Illumina for all the data I showed you here. Yeah. Yeah, no, that'd be great. That'd be great. Oh, cool. Is there any special reason for using the Pseudomonas aeruginosa for the study? So actually, Pseudomonas we used for both. So in both cases, the postdoc and the PhD student who were working to answer these questions had experience with the system. So it kind of goes to the question you were asking before, where if you've worked with a diversity of systems, is there a huge disadvantage in that? And so anybody who comes to work in my group, as long as they're willing and the system they want to work with addresses their question of interest, they can work with, of course, a system where they have previous experience. So in the case of Kim and of Alex, they had previous experience with Pseudomonas aeruginosa. Another reason is that it's a widespread animal pathogen. We were really interested in the impacts of protective microbes against these opportunistic infections. It also has a well-annotated genome, huge implications in the medical field, as well as in thinking about conservation of wildlife and wildlife health. So it was good for all those reasons. And it grows really well in the lab. Cool.
Thank you so much for presenting.
Right.
Yeah, so we have some pizza and snacks at the back. I want to make one comment. If anyone was interested in the studies that Anmol's been doing, there's also some papers there with more info there, or Anmol can talk about that as well. Okay, awesome. You guys enjoy. Thank you for coming.
That's not picky,